Okay, I think it's time to start. So welcome everybody. Today we are going to talk about functionalization of extracellular vesicle. And specifically today we have Phuong, a PhD student from National University of Singapore. We present a paper titled Direct Delivery of Immunomodulator to Tumor Infiltrating Lymphocyte Using Engineered Extracellular Vesicle. It's a paper by Liu et al. From Kanazawa University in Japan. This paper have been published actually this month. It's a very recent paper. And we are going to talk about functionalization of extracellular vesicle for expression of multiple antigen on the surface in order to activate CD8 cells. So we are going to talk about application of extracellular vesicle in immunotherapy. So please, Phuong, you can go ahead, sharing your screen.
Can you hear me well? Okay, good morning, everyone. So welcome to today's Journal Club. As you know, current cancer therapies, including chemo, radiation, and immunotherapy, often lack specificity, leading to off-target toxicity and drug resistance. And extracellular vesicles have emerged as a promising delivery tools due to their excellent biocompatibility and the ability to transport therapeutic molecules directly to target cells. However, their use in cancer immunotherapy have been limited so far, partly because of the lack of essential co-stimulatory signals and cytokines required for effective T-cell activation. My name is Phuong. I'm a PhD student from National University of Singapore. Today, I present this very recent paper, Direct Delivery of Immune Modulators to Tumor-Infiltrated Lymphocytes using Engineered Extracellular Vesicles, published on the Journal of Extracellular Vesicle last month. So they try to address the unmet need in the field by engineering an antigen-presenting EVs that integrate multiple stimulatory signals, enabling precise activation of tumor-specific T cells. And this approach enhances tumor responses while minimizing systemic toxicity, offering a novel and adaptable platform for cancer immunotherapy. Here is the schematic of their work. So they first engineer the antigen-presenting EVs and explore their potential to activate T cells. So these antigen-presenting EVs, which is called FEVs in short, carry 3 immune modulators. The first one shown here in blue is called peptide-MHC class 1 complex. They will carry the antigen peptide, which is ovalbumin, which tumor antigen in a mouse chosen in this study. And the second signal is interleukin-2, which will interact with interleukin-2 receptors on T cells for activation and proliferation of T cells. And the third one is the co-stimulatory molecule, CD8 T, which is shown in purple, which will bind to CD28 on a T cell, which serve as a signal too for T cell stimulation. So they aim to selectively deliver these immune modulators to tumor-specific T cells to promote a robust and targeted immune response. So after generating FEVs from HEC293 T cells, lentiviral transduced with the fusion protein construct, they perform EV characterization. So in the construct as shown on the picture here, they have CD81 fusion protein fused with the single chain peptide-MHC class 1 and interleukin-2 at the extracellular loop. And on the other fusion CD9 construct. They fuse with CD8 T costimulatory signals. So to verify the concurrent expression of this fusion protein, they have here is CD81 fused with RFP and CD9 fused with GFP. So they purify the EV from the supernatant of HEC293 T cells and perform confocal microscopy to quantify the co-localization degree of IFP and GFP signals as shown on the bar graph on the right. So the findings show that about 80% of EVs co-express GFP and RFP, suggesting that the majority of FEVs simultaneously display over MHC class 1, interleukin 2, and CD8C fusion construct. They next investigated if FEVs can stimulate naive CD8 T cells. So first, they isolate over-specific CD8 T cells from the mice, which is also called OT1 in short, and label them with cell trace violet, then co-culture them with various doses of FEVs or control EVs. So the bar graph and the percentage of cell trace violet negative OT1 T cells under different concentration of treatments. So as you know, the T cells will lose the cell trace violet staining upon expansion, resulting in the higher percentage of negative cell population. So after 3 days of cultivation, a dose-dependent expansion of OT1 T cells was observed with FEV treatment as shown by the red bar. This indicates that FEV can directly activate naive T cells. Moreover, when they compare the stimulatory capacity of FEV to a commercial T cell activation kit, CD3, CD28 bits. So after 3 days, they found that FEV was shown to activate OT1 T cell, leading to the production of interferon gamma and granzyme B, which is to a comparable or even a bit higher than the commercial CD3, CD28 stimulatory bit. So collectively, these results indicated that FEVs can directly present antigen stimulatory molecule and cytokine to induce robust proliferation of antigen-specific CD8 T cells. So their aim is to effectively deliver this construct to antigen-specific T cells, which could be safer than administer the drug in the free form, which had been known in the case of freely administered interleukin 2, which might result in a severe adverse effect. So the first, they want to know if the construct stimulate all population of T cell just like the freely delivery drug, or they can selectively activate antigen-specific CD8 T cell only. So to address this question, they isolate antigen-specific CD8 T cell, OT1 cells, and the polyclonal T cell, which is T cell or T cells, and label them with cell trace violet, inoculate them with control EVs or free interleukin 2 recombinant protein and interleukin 2 monoclonal antibody shown in blue and FEV which is shown in red here. So as we can see, the treatment with interleukin 2 recombinant protein and interleukin 2 antibody combo treatment resulting in expansion of both polyclonal T cell and antigen-specific OT1 T cells. In contrast, FEV treatment only triggered the expansion of OT1 T cells. Furthermore, FEV also resulting in the enrichment in the population of T cell, which is high for CD44 and low for CD62L, which represent the effector memory phenotype of the T cells. This population have been shown to have strong capacity to fight cancer cells, even in clinical settings. So having demonstrated functional effects of FEVs, they perform pharmacokinetic study to clarify the therapeutic windows or the functional duration of FEVs. So the mice, recipient mice, will be treated with FEV prior to certain days, or ranging from 0 to 6 days. And then they follow by the adoptive transfer of OT1 T cells, and the BBMC will be collected 4 days for analysis. So this graph here showing the percentage of T cells that are negative for the cell trait violet stain, which is reversely proportional to their expansion capacity. Directly proportional. So we can see after 4 days, there are still 50% of the T cell that have expansion capacity. And so that means their FEV could remain active in the system up till 4 days, about 50%, and then they will gradually decrease at 6 days. So to investigate anti-tumor effect of FEV, they subcutaneously inoculate overexpressing EL4 cells in mice, followed by injection of the treatment, including free interleukin-2 antibody and FEVs, and monitor the tumor growth. So the tumor growth curve represent here include untreated group shown in black, control EV shown in brown, and free drug shown in blue, and FEV shown in red. So we can see the administration of FEV significantly delay cancer progression, and the survival graph here also shown that 6 of 9 mice survive until the end of the study. So just take note, this one is adoptive model because they adoptive transfer the OT1 CD8 specific T cell before the treatment. So in the next model, they also explore the potential of FEV to treat the mice via stimulation of their own endogenous T cell without adoptive transfer. So the treatment scheme is still the same. Tumor inoculation followed by treatment, it just without the adoptive transfer of antigen-specific CD8 T cells. And they observe the similar results. So they can see that the FEV treatment resulting in substantial delay in the cancer treatment and able to prolong the survival in the mice compared to the control group. So they would like to investigate the mechanism underpinning the anti-tumor treatment response as shown in the previous slide. They collected the BBMC from all the treated mice at the end of the study and quantified for the percentage of antigen-specific T cells by tetramer staining. So usually, the percentage of antigen-specific T cell in the blood is very low, often below 1%. So that's why in the non-treated mice here, we can see that only around 0.025% of the CD8 T cell are specific for tumor antigen, and similar for the control group and the free drug. However, in the group treated with FEV, we can clearly observe a dominant, obvious expression of this tumor antigen-specific CD8 T cell, which is around 3.6%, which already considered high for this population in the blood. And they also seen immune checkpoint inhibitor is really emerging nowadays. So they also explore the synergistic effect if they combine the FEV with anti-PD-L1 and immune checkpoint blockage inhibitors. And then they observe a further increase in the percentage of antigen-specific CD8 T cell, reflecting a synergistic effect between FEVs and anti-PD-1 antibody. So since both quantity and quality of antigen-specific T cell are important, hence they continue to investigate the tumor microenvironment after treatments. They label FEV and perform PET scan and found that just like other EVs, the FEV also mainly accumulate in the liver and spleen. However, in the EV receive the FEVs, they can see their accumulation at the tumor site. So, which means there's a potential to deliver the immunomodulator to the tumor cell by EVs. And they perform further analysis on tumor-infiltrated lymphocytes. And so they dissociate the tumors and use tetramer staining to identify the percentage of antigen-specific T cell within the tumor tissues. And expectedly, they found a high percentage of antigen-specific T cells in the tumors of mice received FEVs. And these cells are not just in quantity, they're also functional, as shown by high expression of interferon gamma, as shown on the above graph on the right. And this concurrently accompanied by the decrease in the population of CD8 T cell expressing PD-L1, we see an exhaustion markers. So those previous slides was shown as a proof of concept for mouse tumor antigen. And in the end of the study, they also want to explore if the platform can be customized to treat the human tumors. So they create the so-called humanized FEV version, whereby instead of OVA, they use the NY-ESO tumor antigen peptide, which is very common in melanoma, ovarian, or cervical cancers. And they also use the human version of CD8 T and interleukin-2 instead. So they perform an in vitro study by incubation FEVs and control EVs with human BBMC transduced to express NY-ESO. So after a few days, they observed that BBMC treated with FEV has increased expression level of NUR77, which is a marker of antigen-specific T cell when they are activated, which also accompanied with the increase in the proportion of CD69 T cells shown on the right. And CD69 T cell is the marker for early activation of T cells. And as well as the increase in other functional marker of T cells, such as interferon gamma, granzyme B, as well as the total number of antigen-specific T cells in the treatment. So in conclusion, the study focused on FEVs, which are the engineered EV designed to activate antigen-specific T cells via T cell engagement, co-stimulation, and cytokine signaling. Compared to dendritic vaccines, FEVs offer greater scalability, stability, and customization to overcome manufacturing challenges and cost limitation. Furthermore, selective interleukin-2 delivery to antigen-specific T cells via FEVs enhances safety and efficacy, avoiding systemic side effects seen in the free drug therapies. However, this study also have many limitations. So firstly, I would like to comment on the purity of their EVs because during the purification process, they just simply use ultracentrifugation to spin the EVs without any sucrocushion for gradient density isolation or filtration for size purification. So that means there's a high chance that the EVs are contaminated with other contaminant in the cell culture. And furthermore, that is like in the concept of their study. So this EV will heavily rely on the HLA molecules. In human, HLA or MSC class 1 molecules are extremely diverse. So if they really want to adapt their platform to translate it to the clinical setting, they have to spend a lot of effort and money to develop a library of FEVs which could tailor to hundreds different HLA peptide molecules for broader clinical application. And since their platforms now still rely on lentiviral transduction of HEC293, it could pose a long-term safety concerns. And as you can see in the whole study, the mouse model they use is subcutaneous models. The tumor grown subcutaneously could be very different from the primary tumors in terms of tumor microenvironment, stroma, and even pharmacokinetics. So they have not proven their efficacy using orthotopic model yet. So yeah, that will be all for my presentation today. And thank you for your attention. Now we are opening for any questions.
Thank you, Phuong, for your presentation. And yes, it was very informative. And we can have some questions from the audience. Is there anyone wants to ask something about this paper, Phuong? Please go ahead.
Hi, Phuong. So thank you very much for your presentation. So may I ask the mechanism of like how the EVs can specific target the CD8 plus T cell, and as you show in the slide number 5, so there is like you use polyclonal and with the testing with the interleukin-2 and interleukin IL-2 antibody here, but like because I think in your APEV also have IL-2, so but why in polyclonal here that you don't have any effects there, but only OT1 have. Yeah, so I don't understand on that. Yeah.
Okay, I think let me show back to the schematic here. Yeah, in theory, I do agree with you that the FEV here also have interleukin-2, right? So why they cannot activate the polyclonal T cells? So it lies on this contract with core peptide-MHC class 1. So in this peptide-MHC class 1, they carry a small ball here, which is the OVA antigen. So if they only can bind to the CD8 T cell that carry the T cell receptor that's specific for this antigen. So then this signal will click and bind. And because so in theory, this interleukin-2 can bind to any other cell like polyclonal T cell, for example. But since they have this the MSC arm here, so I imagine they will preferentially bound to the CD8 T cell that have this CAR specificity, and they preferentially stimulate them. And another one is this interleukin-2 alone or MSC alone will not be sufficient to cause stimulation and expansion in T cells. For the T cell to be activated and expanded, we need all three signals. First is from the tumor antigen, second is from the co-stimulatory and co-stimulatory here, CD8 T, sorry. And the third is from the cytokine. So together, these are the three signals required for robust activation of T cells. So coming back to slide number 5, we can see for this here. So they show the preferentially activation of CD8 T cell that specific for the antigen because they have the MSC complex that prefer to bind to the antigen specific CD8 T cell compared to the polyclonal T cells. And however, in the interleukin-2 alone, we still can see the expansion of both population. I think because they are smaller than EV, and then they will have a very strong binding. So the binding, I imagine, is more efficient. So they still able to expand the T cell in vitro. However, this one is very dangerous if we do in the human, because the massive release of activated T cell will lead to a lot of immune adverse event. So yeah, short answer to your question is because of the MHC molecule expressed on the FEV that make them preferentially bind to the antigen-specific CD8 T cell compared to any other T cell that lack of the TCR specific for the MHC complex.
Okay, thank you very much. Yeah, I understand that maybe they will enhance the chance that the ABEVs can meet the CD8 plus T cell, but I still not not clear on how the ABEVs work with other kind of cells. How can they avoid activation of other cells? Yeah. So yeah,
Yeah, maybe it's not enough. But like when they bind to other cells, basically they only interleukin 2 and CD8 T will function, right? They will lack of signal 1, MHC class 1. So they may not. So usually in the immune, if you stimulate the T cells with all the signals and cytokine signals, but without the MHC signals, it will lead to the energy and exhaustion of our T cell as a way to protect our body from non-specific immune activation.
yeah, okay, I understand a little bit now. Yeah, thank you very much for your explanation.
Thank you.
I have a question. I mean, you mentioned it a little bit in the final slide. It's about the human leukocyte antigen, which one of the probably biggest limitation of this preclinical data, and also is a limitation of similar technology. So as you mentioned that the human leukocyte antigen varies a lot among individuals. So it's very difficult that these technologies, as it is, can find clinical application, right?
Yes.
So I would like to know your point of view, which kind of strategy can be implemented to efficiently develop and produce personalized technology where EV is used for antigen presenting in order to overcome the diversity of a human leukocyte antigen. How can we solve this problem?
I think this problem cannot be solved easily because it's really diverse. Even in the same MHC molecule, in the same isoform, they still can present different version or isoform of the tumor antigens because the way the HLA the antigen presenting process the antigen and present it on the MSC. It could be random. So not all tumor antigen the same. So in my many company, including my former company, they also spend a lot of effort how to make this more applicable because it really time consuming if we phenotype each and every patient. You can see in the BBMC, there's only a few percentage or even less than 1% of the tumor antigen specific T cell that you can found to isolate and then to confirm their function before you make the product. I don't think the patient can wait that long. So one of the way is to perform mass screen of the whole population to build a library. So when you have a library of antigen-specific T cell with MHC molecule in place, all the construct in place. So when you screen the patient and okay, this patient is specific for this type of HLA, and this is the tumor the patient express with this antigen. So you will pick them from the library and then quickly make it into a product to deliver it into the patient. So this is the only way that I know for many companies that adapted this, but it will take them years to build up the libraries, to collect the information from the publics, to validate whether these HLA complex are real and functional. Yeah.
Okay, thank you very much for your insight. Yeah, still a long way to go, I believe.
Yeah, but it attract a lot of attention because now with bioinformatics tools, you be able scientists to predict the MSC's class 1 structure and the peptide structure better than before. So yeah, it's an exciting field, not only for EV, for CAR T cell as well,
Yeah,
for
yeah.
MHC, TCR T cell, which are also heavily depend on HLA diversity.
Okay, so if there is no other question, thanks again, Phuong, for your great presentation. And I
Thank
will
you.
see you next week. Thank you, everybody, for joining this section. Bye-bye.
Thank you.